Hello, I'm Maria Puglisi, and my group tested the effect of salinity on the enzyme trypsin. Enzymes are protein molecules in cells which work as catalysts. So catalysts is a substance that increases the rate of a chemical reaction without itself undergoing any permanent chemical change. Enzymes speed up chemical reactions by lowering the activation energy for the reaction. So lower activation energy equals a faster rate. So enzymes are important in living organisms because they facilitate chemical reactions in the body. Enzymes are needed for like metabolic pathways, respiration, digestion, and other important things in the body. Basically, enzymes help maintain homeostasis. And so the enzyme we worked with was trypsin. Trypsin is an enzyme that can be found in the small intestine. So trypsin digests proteins by catalyzing the hydrolysis of peptide bonds on the carbonyl side of the amino acids lysine or arginine. Active trypsin in the small intestine is approximately a pH of 8. So our purpose for this experiment is to determine any salt concentration other than the optimum 0.5 molars will result in a more or less active enzyme. So the reaction that's catalyzed by trypsin, it's the protein azocasein has a sulfonide groups. They are covalently linked to the peptide bonds, and the sulfonide groups are orange. So trypsin is an enzyme that liberates these very short peptide chains and amino acids in the azocasein. So the more short peptide chains and amino acids will result in a more pigmented orange color. So the intensity of the color shows the relative activity of the enzyme trypsin. So the more orange something is, then that means we know the enzyme was being more active. So basically, in animals, trypsin is found in the small intestine, and in bacteria, trypsin is secreted out by them. Some questions we had for this experiment was, what effect would salinity have on enzyme activity? And our hypothesis was any higher concentration of salt other than the optimum concentration of 0.5 molars will cause the enzyme to be less active, as well as lower any concentration will lower the enzyme activity. So essentially we're saying any salt concentration other than 0.15 would result in less enzyme activity. And our reasoning for this hypothesis was because other experiments that we looked at, they also used 0.15 to be their optimum. Okay, so then the materials and methods we used, we had the enzyme trypsin, we had TCA, and we had the substrate azocasein. So the independent variable for this experiment was enzyme activity. The dependent variable is salinity concentrations. And our control variables, we had a pH of 8, temperature of 37 degrees Celsius. And our negative controls, we had tubes with no trypsin, it was just water, and we replicated each salinity 3 times. So our procedures, we started out with, first we labeled 6 microcentrifuge tubes. We had A1, B1, C1, D1, E1, and F1. Each one had a concentration. A had 0, B had 0.075, C had 0.15, D had 0.3, E had 0.6, D had 1.2, F had 2.4. So we repeated this step for labeling for the other 3 applications, so like A2, A3, B2, B3, etc. For each tube experimental treatment, we added, the 0 for tube A, 0.75 for B, 0.15 for C, 0.3 for D, etc., etc. And then we added 500 microliters of azocasein to all 6 tubes, A through F. And then we added 50 microliters of trypsin to all 6 tubes, A through F. We incubated for 20 minutes, so like temperature 37 degrees Celsius, pH of 8. We added 500 microliters of the TCA. And the reason why we added TCA is because TCA stops the reaction, and it precipitates macromolecules, including the enzyme and undigested azocasein. So that's why we add TCA. And after you add the TCA, you place all 6 tubes into the centrifuge, and we centrifuge it for 2 minutes at 14,000 RPM. And the reason why we centrifuge it is because We centrifuge the reactions after TCA in order to remove undigested substrates, very short peptide chains, and free amino acids liberated from the trypsin are not precipitated by the TCA, so thus they will remain in the solution. And then after centrifugation, we transfer the supernatant to the new tubes, and then we have 6 new cuvettes labeled A1 to F1. So we take the blank cuvettes and we place it into the sample holder, close the compartment door. This is for our spectrophotometer. We press the automatic blank key to set the blank to 0 absorbance. Remove the blank and you insert the cuvette holding the supernatant. And then we close the compartment door and you read the absorbance at 440 nanometers, okay? And after that you read the absorbance, you record it, and you repeat these steps for the other supernatants holding B1, C1, D1, E1, F1. And then you repeat steps 2 through 17 for all the other replicates here. So basically the substrate we used for this experiment was azocasein, which is a chemically modified protein that was designed as a substrate for, for finding out enzyme activity. Azocasein is prepared by chemically adding the sulfonide groups to the milk protein casein. So the sulfonide groups, which are orange, are covalently linked to peptide bonds. Therefore, the intensity of the color measured by spectrophotometry at 440 nanometers is used to determine the relative activity, of the enzyme trypsin. And so with our negative controls, we use a negative control because we're supposed to produce no result from it to make sure that what we're doing is correct. So our negative control again was tubes with no trypsin. We had just water in them. And the reason why we have replicates, had 3 replicates. The reason why we have replicates is because it's to produce more accurate results, to see if there's any outliers in the data, and it reduces variability in the experiment. So our results from this experiment, we found that tube A, which was the 0 concentration of salt, had the highest enzyme activity because it had the highest spectrophotometer reading, therefore it had a more intense color. And tube G, which had a 2.4 concentration of salt, had the lowest enzyme activity because it had the lowest spectrophotometer reading, which means it had less color. So basically, lower salinity levels result in a higher spectrophotometer reading, and higher salinity results in a lower spectrophotometer reading. So this is the graph that we got for that. You can see A had the highest absorbance rating, while G had the lowest absorbance rating, and then these lines show the standard deviation between them. So basically, we see that, so when we were comparing our p-values with the results for the null hypothesis, the absorbance at salinity 0 is not significantly different than the absorbance at 0.075. So the result is not significantly different. The absorbance at salinity 0 is not significantly different than the absorbance of 0.5. It is significantly different. It's basically significantly different for all of them compared to 0. And basically, when we test standard deviation measures the amount of variation in a set of data values, so it's just comparing the deviation in the single set. When you're doing p-values, it's comparing the deviation kind of between 2 different sets. So that's what this result means. And then this chart here shows all of our results, and the numbers gives our normalized average, the standard deviation, and it shows what our controls were too. So yes, our results, everything ends with the lower salt concentration results in higher enzyme activity, higher salinity results in lower spectrophotometer reading. So our conclusion for this experiment, we can say that the experimental design and execution was adequate to test our hypothesis. So our discussion/conclusion is, after looking at our experiment's results, our hypothesis was proven incorrect because with lower salt concentration, there was higher spectrophotometer readings with higher salt concentrations. So there were lower spectrophotometer readings. But because we performed multiple tests of each concentration, our results should be reliable. So we predicted that both high and very low salt concentrations with lower enzyme activity. However, low salt concentration increased its activity. So half of it was right, half of it was unright. So we have to say our hypothesis was proven incorrect. Also from the student's t-test, we saw that the salinity concentration at 0 is not significantly different when it's like compared to 0.05, but then for everything else it is significantly different. Okay, so from our conclusion, we can say that the other groups can both support and contradict our data, because the other groups found that 0.15 was the highest salinity concentration for that, like the highest enzyme activity for that salinity concentration, but an increase in that also equals a lower enzyme activity. So all of our groups found increasing the salinity results in lower enzyme activity. However, another group that tested salinity found that 0.15 was their highest. So our choice of control variables resulted in the highest possible rate of enzyme activity, because these are the conditions in the small intestine where trypsin is. So like our control variables again was pH of 8 and temperature 37 degrees Celsius. We chose pH of 8 because that's the pH inside the small intestine, and 37 degrees Celsius because that's the body temperature in the small intestine as well. Sources of variability for this experiment could be, maybe we had a rushed incubation time because we're running short on time, or human error with like measuring amounts. Someone could have not pipetted the right amount into something. And so potential improvements for the next time for this experiment is maybe we could test like even a more variety of salinity concentrations. Maybe we could have more replicates. And the relevance of this conclusion in terms of ideal condition to grow bacteria is environment with the lowest salinity should grow bacteria the best, because lower salinity produces a higher enzyme activity.